sentence in an abstract to a paper that you submit to a prestigious journal.
What?
That most of the stuff is obvious and bits that aren't obvious.
Yeah.
Sounds like a referee report.
You can quote me on that. Yeah. That's my son. He came on the hike with us, made us walk fast. All right. Disclaimers. Additionally, this is a progress report about something, and I don't know what I'm talking about. I want to make that clear for any viewers at home. You can't actually ask me about any of this. All right. So I work in theoretical immunology, and I got this quote last week, and this is one of my favorite quotes about immunology. So when they originally split lymphocytes into T cells and B cells, which they didn't do until 1968, they were accused of complicating things, including somebody from Australia, an Australian being Stochastic, said that B and T were the first and last letters of the word bullshit.
Really?
Sorry, yeah. Anyway, I put this up there because it's kind of, I just want to let you know that immunology is actually a new science. There wasn't a great deal known about immunology until the 60s. And in my lifetime, a whole theory of immunology was built and destroyed, really destroyed down to the ground. And there aren't too many sciences where you could actually say that. All right, so in today's talk, I'm going to talk about B cells. I just want to show you at least a picture of a B cell so you kind of know what we're talking about. This would be a non-activated B cell. You would have lots of these in your lymph nodes and in your blood. And on the surface of the B cells, there are B cell receptors, and these are basically surface-bound antibodies. Okay, so if you know an antibody that will stick to something, B cell receptor is a surface-bound antibody with some additional signaling machinery associated with it. And it will bind on to antigens and pathogens. That's all we need to know for today. Okay. Later on in the talk, there will be a brief mention of these things called toll-like receptors. So toll-like receptors are generic, and they'll bind, for example, to lipopolysaccharides or this kind of DNA, and those are both signatures of bacteria. Okay. So B cells are also able to detect just when there's generically bacteria around, as well as when there's specifically the antigen that they recognize. Okay, and if you stimulate B cells with TLRs, they might look something like this under electron microscopy. They actually look different. They get activated and things work differently. I'll be coming back to that later on. All right, so we have these receptors on the surface of the cell, and we've got tens of thousands of them. Okay, so there are tens of thousands of identical surface receptors of this kind that I call BCR on a given B cell. Okay, and if we do TIRF, which is total internal reflection fluorescence microscopy, we kind of see this kind of blurry image, but you can see it's patchy. The distribution of receptors is certainly patchy. If you do STORM at some arbitrary resolution, then you see that the patchiness is actually, there's true patchiness here in your STORM data. Okay, and you can also track them. So,
you remember the length scales?
Yeah, just a second. Yeah. You can also track the receptors. So this is actually our data. I'll get back to why I don't like this data so much, but you can see the little beacons here. These are quantum dots which are attached, hopefully monovalently, to B cell receptors, and they're wiggling around. All right, length scales. We'll go with like 5, 6, maybe 5 to 10 micrometers across this guy. This would be one cell sitting here on the glass. So these guys are moving around with diffusion coefficients, which are at the fastest a micrometer squared per second, at the slowest probably a tenth of that, down to immobile. Okay.
So shouldn't they be called microclusters rather than nanoclusters?
Well,
It just sounds sexier to call them
these guys?
nanoclusters.
These guys are nanoclusters. This is STORM microscopy, so you can zoom in on this down to here. This is a zoom-in image of that square. Okay, so this really is just about 20 nanometers or 50 nanometers or something. Okay, and we're taking that data as well these days. Yeah. So no. Okay, the general goals that I have in my life are to probe B cell activation through particle tracking and doing super resolution STORM experiments, which I'm not going to talk about much today at all. Characterizing clustering of the receptors and then understanding how that builds into signaling. And the sort of long-term goal would be to build a spatiotemporal signaling model for B cell receptor. I actually don't think I'm ever going to do that, but that would be kind of the big picture. All right. Okay, but the menu for today, I want to talk in a very basic way about single particle tracking experiments and theory, talk about the technique, talk about how you're going to fit a diffusive model from this, because there's a lot of strange misconceptions in the field, I think, and how you might look for confinement in single particle tracks. I want to talk about B-cell receptor control, the motion control by actin, and a mixture of failure and success refers to our ability to study this experimentally. And then I want to show a preliminary study on clustering of B-cell receptors that we're doing in the lab. And I have a little bit of analysis of that, but that's one. I'm hoping to work on parts 1 and 3 while I'm here. So that you might find me working on these. Okay, all good? All right, so I'm going to talk about this is how we do single particle tracking. Okay, so what we've got is we've got a 3-color turf illuminator. A turf illuminator shoots the lasers in at the correct angle. Here they come in so that they undergo total internal reflection in the glass, and you get this evanescent wave which only penetrates a few tens of nanometers into the sample. Okay, so you're only illuminating. You can tune this by tuning this angle here. You can tune how deeply you penetrate into the sample. So that's a parameter that you can just type on the computer, and it controls this illuminator thing, which sets this in. All right, and then we've got 2 cameras, so we can split, for example, green and red here, so that we can image 2 different colors. Okay, so we can actually input 3 colors. Right. We can image 2. Okay, so this is how the microscope basically works.
Sir, when you say image different colors, that's not the actual colors of the thing. That's you're using the light to probe at different lens skills, or what?
Well, if I have a green and red fluorophore, right?
Okay.
Okay, which I will have later on. Right. Okay. So how are we going to label? Okay, so the 2 ways that we've been doing the labeling. The first way is Fab labeling. So Fab. This is an antibody, right? It's got 2 chains, unless you're a camel, in which case it only has 1. That's actually true, or a llama. But you got this thing. You digest it with pineapple enzyme into 2 different bits like this, and then you attach, in our case, a Cy3 dye. Okay, so this is a cyan 3 dye. Okay, and you saw the movie here, and the issue with this is these things tend to bleach out. Okay, so basically you get a lot of short tracks with your site with this. Okay, we do the best we can. We use the lowest laser power that's consistent with getting good signal to noise, enough signal to noise here that we can track. Okay, but they tend to bleach out. So typically, a typical experiment here, we'd see tracks. A few of them would be long, but a lot of them would only have maybe like 10 frames or 20 frames. Okay, so they're not terrifically long. Okay. Yeah. All right. That's interesting. Okay, the alternative, I think that's going to be a movie on the next one. The quantum dot labeling is a little bit more difficult. So the benefit of quantum dots is they don't bleach, so you can get long tracks. So we were all gung-ho about using quantum dots a few years ago. So basically, again, you get your Fab. This time you put biotin on it. Biotin binds to streptavidin, which is on the quantum dot. Okay, and you also block to try to prevent multivalent attachment of the quantum dot to the cell surface receptor. Okay, so you try to do this all at limiting dilutions and with blocking, and by blocking, try to prevent multivalent attachment, but you can never completely rule it out.
What about Q dots makes them not bleach?
Q dots, though they're little bits of semiconductor. They blink, they don't bleach. They blink, they will switch off for a while, but they'll usually come back. Yeah, so on the left here, we have a movie of Fab Cy3 experiment, and you can see kind of dimmer things moving around. And on the right, we have a movie of, which is apparently not going to work. Okay, that's great. I don't know why that is. Yeah, it's a green screen, yeah. Imagine some sort of brighter objects. Let me try that again. No, it didn't work. Imagine some brighter objects moving around, but not as much. Okay, all right. Sorry about that. That was working in my office. All right, and it also worked at Oxford, so I don't know what's up with the projector here or whatever. Okay, so you take your movie, you store it on your computer, and now you need to extract the tracks from the movie. Okay, so we don't do that, by the way. We buy software. We get free software that does this. This is a problem which has been solved many times. It's not one that I'm interested in solving again. Until recently, we were using a thing called U-Track, which came from one of the labs at Harvard. We've now changed because we think IC is better. Okay, We find better particle detection, which is the first step, and then we find better joining. I'm not actually 100% sure that joining is really better than U-track, but the detection is definitely better.
What do you mean by joining?
Joining each frame of the movie will have multiple points in it, and then you don't have to join them together to make tracks.
Right, I see.
Okay, yeah. If you only had one, you wouldn't really have to join, but with multiple tracks you do. And the better particle detection really allows us to use Psi3 much more confidently. Psi3, the signal to noise, is not so good as the QDOTs, and so we're doing better with a lot better with the IC. And since we started using IC, we've become a lot happier about using the Psi3. Okay, all right. So that's kind of the practical side. So then you imagine a spreadsheet with each of your particles and each of your positions. You see particles going across, your positions going downwards. So what are you going to do with that? Well, this is a question that's been bugging people for a long time. What we're going to do with that is I just want to talk about Brownian motion. So nothing complicated here. All right, so let's just assume that the steps, which you observe in your track, are drawn from Gaussian distribution, okay, with the correct variance. All right. This is a self-similar thing. So, saying Brownian motion is basically saying that on any length scale I want to look on, I've got this scaling between dx and dt. Sorry. Did I get that the wrong way around? Yep. Yeah, that should be dx squared scales as dt. Sorry. See, I really don't know what I'm talking about. But I knew that. That's just a mistake. Okay. And in principle, this is valid above millisecond time scales. All right. If you go down below that, then there's all kinds of very high resolution experiments that show that it's not a good model below that. Okay, so challenge then, I want to say, is let's fit pure diffusion to single particle tracking data and understand the deviations. All right, so I know how to do the first part. What I don't know how to do necessarily is how to do the second part. So how to understand the deviations from a pure diffusion model.
Remind us, you're tracking receptors on the B cell?
Yeah, single receptors on the B cell. Yeah.
This would be surface diffusion?
Yep, they're on the plasma membrane of the B cell. Yeah. Okay, so how do we do that? All right, so here's a simulated track. Okay, so here's a simulated track. And what I'm going to say, this is the traditional approach. The traditional approach is plot the mean square displacement as a function of time. Okay, so you do something slightly more subtle than just plotting the squared displacement over time. You take every possible subtrack of length 1, and then you average those, and that's your first point. And you take every possible subtrack of length 2, you average those, and that's your second point. Okay? So basically, there's a lot more, there's n points going in here, and out here, there's a lot less than n points being averaged. Okay, and that accounts for this noise that's showing up at the end. Okay. And then you plot this against the number of points in your subtracks. Okay, and you expect that the slope of that should be 4D.
That would be if it were in a two-dimensional plane. The fact that it's on a two-dimensional curved surface, does that change that?
Yeah, we can always ignore that. Everything's 2D for today, yeah.
If I plot sort of non-independent sort of plot, sort of artifacts over here, appear over here as well, because the same data being used over and over again?
Yeah, that could happen. I'm not promoting this.
Right.
I think it's a terrible way to operate.
Yeah.
All right, but I'm telling you what people do. Sorry, I should have made that clear. Okay. Okay, so you fit a straight line through that. And best fit is 1.14. So by the way, I simulated this with d equals a half, and I got d is about a quarter, which is just terrible. And I didn't cherry pick this one. This was the first simulation that came out. All right, actually it's the second one. The first one gave a worse estimate because it was even less linear than that. Okay, obviously there's a lot of variability. Okay. But what you can do is, if you say, okay, there's a lot of noise out here, so I should just fit the first few points, right? So what I did here was I just averaged the first 10 points and fit that, and then I get d is about 0.43. Okay, so that's better. All right. Now, if you look in the literature, back in the days when there weren't very many single particle tracks around, things like this and this were enormously appealing to experimentalists and also some misguided physicists. You know, a downward-facing curve here was interpreted always as anomalous diffusion. It just isn't. Okay, you cannot make that statement. Right? You'd have to have a lot of data to make that statement. Okay, so here are 5 tracks, And you can see some of them have downward curving MSDs, and here some of them have upward curving MSDs, and some of them don't really go anywhere. You know, they end up not going anywhere in the long run. They kind of just hang around 0. So what are you going to do with that? Okay, so just
data or simulation.
that was all simulation. This is all simulation. If I use data, I don't know if it's Brownian diffusion or not. Well, you know, so I have to use simulations. All simulations. So if I take 50 of these and I plot all of the slopes of the MSD curves taken over different lengths of windows. So this is the MSD curve with 1 point. So this is just the squares of each of the things. So basically the sum of the square displacements. Right? And this is out to taking like 50 things. Okay. Then, sorry, is this graph clear? Does it make sense? Yeah. So this is a very short time window, and this is a long time window. And basically, the mean is actually pretty good. So I got 50 tracks. So the mean on both ends is good. But which one you want to use to estimate? Well, you don't want to use this one because it's got a huge standard deviation. You want to use this one. Okay. So basically, the single step mean square displacement, if you like, is the best estimator of D. Okay. And I can also show that through maximum likelihood. But I don't really want to go through the details. But basically, if I work out the likelihood of a given diffusion step from a single observation, okay, and the overall likelihood is just going to be all of these things multiplied together. Take the log, I get a sum, differentiate that with respect to the diffusion coefficient. I get this, set that equal to 0 because I'm looking for a maximum, and rearrange a bit, and I get that my maximum likelihood estimate of D is exactly the one-step mean square displacement.
Hang on, that MOE is based on assuming the steps are all independent.
Yes.
Right, so if you had a process which was diffusive but you were sampling at a higher rate than the diffusion time, then that might not be optimal, right?
Yeah. There's lots of places this might
Now
not be
you're
optimal.
saying even if the data really is diffusion, it's hard enough. to measure.
Yeah. I think there's an important thing here is everyone could always say this only applies for pure diffusion, but of course it only applies for pure diffusion. You know, we have a hard enough time agreeing, when we've got one model with one parameter, we have a hard enough time to agreeing on that we're fitting it. As soon as anyone talks about it, everybody wants to, I'm not picking on you, Peter, but everyone wants to talk about everything else.
No, I'm just saying,
yeah.
I mean, physical Brownian motion is
Yeah.
always, even if you just have whatever it is, idealized physical Brownian motion,
Yeah.
there is a correlation time before the diffusion kicks in.
Yeah,
So-
absolutely.
And I'm just saying, I
Yeah.
think what this argument tells you is
Yeah.
that your sampling should be sort of determined by that correlation.
Yeah. There's a correlation time, which I'm not talking. On the next, I'm going to say something similar, but it's not the same thing. There's a correlation time. Fortunately for me, my camera, even though it cost more than my car, actually more than all my cars put together that I've ever owned, it still cannot sample fast enough to worry about that. In assuming pure Brownian motion. Now, if the thing's on a molecular motor, then we'll have to talk. But yeah, yes.
So is this basically a consequence of the law of large numbers, that you've got the most data for those short timescales? Is it just purely because of that?
Yeah,
or
I
is there
think
something-
ultimately, I think also there's a point here that you get kind of one piece of information per time step, and just by resampling and averaging that information, I don't think you can really get around this. So one thing people sometimes say is, well, this allows me to average across noise. So I'm taking several steps because it's sort of noisy, so I should be averaging. That's bullshit. Excuse me. That's B and T cells too. Okay, and so if you add noise, so if you do the maximum likelihood calculation where you add noise, so what I did was I said there's a standard error, error of standard deviation S in X and Y, so I had an X precision and a Y precision. I don't know why I did it like that. Okay, and you do the likelihood. So first of all, your single step probability distribution is convolution of a Gaussian with standard deviation S and a convolution and one with 4D tau. Okay, so then you've still got something that fundamentally looks the same. You multiply them all together, take the log, ... You calculate that your maximum likelihood error is, in the end, the diffusion coefficient you would have calculated anyway, with a correction which scales like this. Okay, so this is the maximum likelihood estimate for D, given some Gaussian noise in the positions.
In practice, is it reasonable to assume S is known?
I'm going to show you how I count. So what you can do now, which is kind of fun, is you stick your particle down, so it doesn't move. You get some, we just dry them actually. We just dry them and they stick. Okay, so now they're stuck. okay? These movies are obviously going to be a problem, but you stick them. Okay, this was a movie of a particle not moving, except that when you look at the pixels, it does move, right? The pixels jitter. Okay, so you get some apparent motion. Okay, and the apparent motion, if you say that, so if you rearrange this thing, you say, well, actually there's no motion, so my MLE should be 0. This allows me to estimate basically the spatial error S. Okay, so it's the square root of 2 times the time step times the motion, apparent motion of a stuck particle, and you square root the whole thing. Okay, and so I can do this. Let's see. No, it's still green. Okay, so I think this is because I'm using the HDMI cable. But anyway, the other one wasn't working. All right, so did anything change when I did that? Oh, no, it's the same slide. Yeah, okay, this is the next slide. All right, so we actually did this with 3 different things. So we used a 1 micron bead, we used a Q dot, and we used a fab. Fab is small and dim. The Q dot is a bit bigger and brighter. The bead is huge and really bright. Okay, and so you can estimate using this basically the positional accuracy of your imaging system and your particle tracking system, your particle detection and tracking. Okay, and we get this nice result that we have 14 nanometer resolution for big, bright objects, and for the objects we're actually using, we have still pretty good resolution, really. This is a nice thing to be able to do. Everyone clear on that? Yeah. Okay, and you can also then say, well, if I think the diffusivity is about 0.1 micron squared per second, which is roughly what I think it is, most people would agree with me on that from lots of different labs, then basically 5,000 frames per second is the point at which this error would be comparable to what you're observing. So you really shouldn't be sampling at 5,000 frames per second for fabs. But fortunately, fabs are nowhere near bright enough, and my camera's nowhere near fast enough for this to be an issue. Okay, all right, we start to hit signal to noise problems at 100 frames per second, not 5,000 frames per second. We just can't go that fast. All right. Okay, so the next thing you might want to ask about, and people have asked about with particle tracking, is confinement. Okay, so this is the original confinement due to Ken Jacobson, and it's sort of been hanging around. Other people have published similar ways of looking at confinement in particle tracks. Okay, so again, this is a pure diffusive track, the same one that we were looking at before. Okay, and one thing you can do is basically you can take a windowed mean square displacement. So you kind of crawl along your track and you try to figure out, is the mean square displacement in this window bigger or smaller than what I expect? Okay, so I did that for this track. Remember, this is pure diffusion. There's no confinement here whatsoever. So depending on the length of my window, by the way, the red line is what is the diffusion coefficient, basically. That's what you would expect it to be. If you look on short distances, of course, you get fluctuations where the particle moves a lot more than it really ought to, but then you get a lot of times that it moves less. This is on a log scale. Sorry, should have mentioned that. It's on a log scale. And then if you increase the length of your window, eventually everything averages out. Okay, so this is a short window, medium window, long window, I guess. Okay, and we're looking at the window mean square displacement. Okay, this is clearly a bad method because you've got to set a window length, and then you've got to set a line on here, which is where do you think significant deviations from normality exist? Okay, and that's a bit of a tricky thing to do. Okay, so I don't like this method. Is that clear? It's clear to me, but...
So confined here does not mean this is a confined region, this diffusion within. This is a windowing.
This is windowing, and you're looking for deviations which suggest the particle's moving sort of less than average, which would be a signature of confinement. Or it could be transient slowing down. That would be another option. Okay. The second one, this one's a little hard to explain. Basically what you do is you take a track and you set a radius, and you calculate over all sub-tracks the mean time, the variance of the time to escape from a disk of that radius. Okay, that makes sense? And then you average that. Okay, so basically what you're computing is the variance of some sort of expected variance, I guess, of the time to escape from a disk of radius r. So here's my track. Here's various radiuses r. Okay, and the variance is biggest around here. Okay, and then you look at that and you say, well, that's r equals 10, and the whole track here is kind of confined in a region of radius r equals 10. So the variance to escape, the variance of the time to escape from a region roughly the same size as the whole track is going to be the biggest variance. Okay, all right. There's very little variance to escape from small regions because that tends to average out and be those numbers tend to be similar. Okay. All right.
Then why does the variance go down as you make r bigger?
Well, once you go past the peak,
Or
because then it tends to never escape in the whole track. Once you pick a big enough r, it's impossible. There's no variance because it's sort of infinite time to escape in all of them. Yeah.
then you're getting like no samples?
Yeah, your number of samples goes down. Yeah. Okay. So I did this instead of for one track, I did for 5 tracks. Okay, so these are 5 random tracks in the top line, and you get plots like this. And you know, so the sort of characteristic behavior here is that you have sort of a peak, which is roughly the scale of where the track is. Remember, this is pure diffusion, right? I didn't add any confinement here, but it's quite difficult to interpret. You know, you get spurious bumps and wiggles and things showing up here. We could probably smooth this out a bit, but again, it's an ad hoc method. It actually came from ecology, and in ecology, they put a collar on a moose, and then they have, I'm not kidding, they put a collar on a moose, and they have not infinitely long, they have an extremely long time series of where the moose is located. All right, so they apply this to try to get some idea about, is the moose sort of confined or is it sort of free in its motion? We, on the other hand, only have tracks of length 20 or something. And it is notable that the one time this has been applied in biophysics, it was with this bead data where they have like 5,000 frames per track, which is a whole different problem than what we're working with. So this one may or may not be a good idea, but it's not very useful for the kind of data we're typically generating. Okay. I almost don't want to talk about this one. You know, for a Gaussian, you've got the second moment is whatever it is, and then the third moment is 0. The fourth moment is whatever it is, the sixth moment, the 8th moment. If you take the moments and plot them against the time step squared or the time step fourth or the time step to the 6th, you should get, it should be the slopes, and then you take the slope of each of those plots individually and plot that, it should be a straight line. And if it's not, that means that some of your moments are either bigger or smaller than they sort of ought to be. And this is deviation from normality. And the reason I mention this one is it's implemented in UTrack, which we're using. And so people occasionally just use this and just say, well, and UTrack says it's confined or says it's free. And then they just label in blue the tracks that are confined and in red the ones that are free. And you will see this around. I haven't verified this method in any detail, but I really don't like the idea. I would be happier just plotting a histogram of your steps and making some comments about that rather than doing all this malarkey. It seems to be just obfuscating the issue. Okay. Yeah, this is what we did now 7 or 8 years ago. Basically what we did was we said, okay, you've got your fluorescent particle and you're observing it, but in fact, there's a state that you can't see is whether it's in a free state or a bound state. So this is probably very similar to what John talked about the other day. So basically, your particle's in the free state. It's drawing its mobility. It's drawing its steps from a wide Gaussian, and when it's in the confined state, it's drawing them from a skinny Gaussian, and then it'll transition from free to bound, and then back to free again, and it'll start moving in long steps again. So what you can do then is you can take your track, and you can basically fit it. This is sort of shorthand for the model. You've got 2 different diffusivities, and the particle is transitioning between them potentially every time step, and you can infer these 4 parameters of the model from the tracks. Okay, so this is what we did. We do this in a spatially independent way, so it's quite different from what David's been talking about in his class. And obviously, you kind of require that your transition rates are sufficiently fast that you find some transitions, but not so fast that you would have multiple transitions per frame of your movie. So you've got to be kind of in some intermediate regime to actually get anything from this. We do this through a maximum likelihood framework, and I don't even want to go through this slide, but basically it works great on simulated data, and when we apply it to biological samples, we get results which are sort of interpretable from a biological standpoint. So generally, we're pretty happy with this method. You went through this on Friday, right, John, to some degree, yeah. Okay.
So here you use this on the data. So this assumes you have some idea, if you have confinement, what it can be due to. Because in our case, in neuronal cells, what we observed is that we had confinement because there was potential wells with, for example, different depth. Now, if you use long trajectories here that spend sometimes in a well with a certain depth, and it can also spend times in another well with a different depth, and then you merge this. I mean, because you see, it can spend time in a small well, in a big well. So here, when you use the same trajectory because it is long enough, it can bind to these two, which should in principle be different. But here what you are saying is, I don't care. Possibly the difference between the wells might not be that big, have more or less a certain homogenization of the wells, they're more or less the same. And so I'm going to put them like one which is representative. And this is the underlying model that you have about binding on your B cells.
Yeah. So what we didn't want to say was we didn't want to try to map out where are the binding sites. So we can actually go back and run the model in reverse and take a track and figure out where it's fast and where it's slow. We can do that, but we don't put too much emphasis on that side. We think there are binding sites kind of everywhere. This is the model.
Okay.
And that the particles bind and unbind kind of in a time, it depends in a spatially independent way. Yeah. So there's no explicit space here.
But this is very strong assumption or strong.
Oh yeah, I absolutely agree.
Consequence on the data. This is no, we don't observe this in neuronal cells. If this happened on the B cell, this is very profound.
Yes.
There's
So
something
the data we've done this on is actually T cells, but it, you know, we've applied this to B cells now, but that's not ready to show. But the original data was T cells. It was long tracks, and when we disrupted actin, we saw changes in the transitions, and we interpreted those because actin is more or less everywhere. We interpreted those as the particle can bind and unbind from actin in different ways. That was basically the thing. Whether that's correct or not, I don't know, but I think it's a good method. I think
How are you reading this nice little figure? I got quite. Actually, just go through it for me.
Yeah. All right. These are three. Yeah, this is a horrible thing to do now. These are. God. Okay. We took three sets of simulated data. Okay. We did the old-fashioned way of analyzing it, which you sort of have a map, just one diffusion coefficient for the whole thing. Okay. And we found. So you get this histogram of the macroscopic diffusion coefficient. And what people had done up until this point is they've taken these histograms and they've broken them into two normal distributions added together. And they'd said, well, there's kind of a slower one than the normal distribution. It's kind of a faster one. And they add them together and you get something kind of looks like this histogram. So this is a sum of Gaussians fit to these histograms, okay? Right. And then this is our fits for D1 and D2 using this method. So remember, we simulated the data using D1 and D2 with some transition rates, which I'm not talking about, and we get very good, with simulated data, extremely good reconstruction of what the D1 and D2 are, okay? And in the case, this middle case was where it was actually just D1, there was no two states. The two states were the same. The model collapsed properly down to one state, as it should have. If I'd included the other panel of the figure where you have the transition rates, generally the estimates on the transition rates are a bit noisy because you really need a lot of transition rates to get a good estimate of how quickly they're happening. We don't usually have enough in our. We could have made our simulated data longer, but we made it the same as the experiment.
Just a figure indicating that case where you really just had one diffusion coefficient, there's a little bit more uncertainty in the estimated parameters.
Yeah, basically the the likelihood surface for the transition rates becomes completely flat because you can't distinguish the two states. Yeah, yeah. And if the parameters are close together here, then the transition rate is fairly flat. If they're a little bit better separated, the transition probability landscape gets more peaky, and you can more easily find the top. Yeah. Okay. All right. So that's the end of my warm-up. So it only took 40 minutes to do the warm-up on particle tracking. I just want to take you through the highs and lows of my attempts to do experiments now. Okay. So I'm going to talk about, oh yeah, this is one thing I want to work on while I'm here, is actually all of these confinement algorithms. I see them as confinement algorithms. I think it would be nice for me to understand how the strengths and weaknesses of the different ways of looking at this problem. Okay. This is quite different. This is purely temporal. This is not spatial. Okay. So I'm thinking about doing that while I'm here. Okay, now for some biology. So if we can just change gears. So what do we have? We had fitting diffusion, we had looking for confinement. I'm basically just going to talk about fitting diffusion for a bit now. Okay. All right, so this is some experimental data from about 5 or 6 years ago. Okay, so this is an image. This is the surface of a B cell. Some receptors are labeled, and you can see the particle tracks of the receptors. Okay. Also, you can see this kind of blotchy, cloudy thing behind. This is actin. Actin has been labeled at high density with a thing called lifact, which sticks to filamentous actin. And this is like a mask of there's dense actin here, and over here there's not very much actin. Okay. And the tracks which are in the dense actin regions are labeled in red, and the tracks which are in the low actin regions are labeled in yellow. Okay. So they just did a binary mask between low and high and labeled them. Okay, so this is now zooming in on this square. You can see particle tracks in the high actin regions and particle tracks in the low actin regions. Okay, so they just did a binary cutoff on the actin density. All right, and then they say, well, inside the actin, we have diffusion coefficients like this, and they're low. And here on the outside of the actin, we have a wider range of diffusion coefficients with a higher average. And there's 3 stars here, which must mean something good happened. Okay, and this plot is basically taking these stack diagrams and replacing them with kind of a smooth curve. Okay, so basically on the inside, you see you have relatively, well, it's basically that stack drawn again. Okay, so you're drawing that, you're kind of folding that over into a frequency plot.
And you estimated the diffusion coefficient by this one-step process that you did?
This is not my work.
Okay.
This is from Facundo Batista. They don't tell you how they estimated the diffusion coefficient, but I have contacts. Okay, and yes, they did it. They did it in a reasonably good way. I think they did a short step. Yeah.
So 0 isn't really 0 here.
0.
There's actually some significant scales there. I mean,
What do you mean?
I mean, it looks like, so those the values on the y-axis.
Yeah.
So that says 0, right? For that bottom set of.
Yeah, like here. Yeah.
Yeah.
Yeah. Those are probably They're probably not moving.
They're not moving.
Well, I mean, they might be moving a bit, but they hardly moved, right?
Yeah.
Yeah, and these guys are moving quite a bit.
Yeah. So there's actually a larger separation of scales that would be suggested by that linear scaling and so on. So.
Yes. Yeah, I think that's right. Just to comment on the numerical values, this is way lower than we ever get. We don't know why, but just difference. Their cells must be different than ours, aren't they? Okay. So they got this result about actin. Basically, the big result here is if you've got actin present, you diffuse slower. All right.
So, for example, they did removing a drug to remove actin?
Yeah, that's the next slide.
Okay.
Yeah. Okay. So they did exactly what David just suggested, and they did not 1, not 2, but 1, 2, 3 drugs to disrupt actin in different ways. And they consistently found that this is the control. Okay, they consistently found that the diffusivity went up, at least the average went up. There's still a lot of non-moving things all the time. Okay, and this is again these plots kind of flipped on their side. All right, David. Good. Yeah, it's all right. It's easy to be a biologist in this area because all you do is whenever anyone says action, you say, "Did you do a cytokilasing experiment?" And you can sound clever. Just learn those words. Did you do cytokilasing? All right. So anyway, okay. So here I come with my microscope, which cost less than my house, but a lot more than all my cars. And so we're getting started, and we decided we wanted to reproduce these results. Okay, so interested in B cells. This is a B cell result. interested in actin. This is an actin result. So we took this and we're going, okay, we're going to try to see if we can get this to go in our laboratory. Now, somebody, somewhere along the line, convinced me, so this is the Batista Labs experiment, it's my experiment. Somebody along the line convinced me that we should use Q dots because they're bright and they never bleach and we could get really long tracks. Okay, so we're basically, they did it with their fabs, we're going to do it with Q dots. There's a few other small differences, like they have the slips were coated with fibronectin and ours are coated with anti-MHC2, but that's not supposed to make them different. Okay, so we did that, and you would see some experimental data there, but that's not going to happen apparently. It's all right. It's just oh.
You can get stills, you just can't get movies, I think.
Yeah. Okay.
Does it show right on your screen?
No, it's showing green on my screen as well right now. It's fine in my office. If you want to come by and see like dim dots moving around on the screen, that's absolutely fine. Okay, so then we fit the diffusion coefficients. And just by the way, because we optimize a lot of stuff, we can get a lot of tracks. We can do a day of experiments and get thousands and thousands of tracks. So our statistics are always going to be, anything I want is significant at this point, whether it's a good thing or not. Okay, so here is my diffusion coefficient from the one-step model. Here's my diffusion coefficient distribution, okay? And here's my immobility threshold. This is actually bad because more than half of my dots are actually immobile, according to the threshold.
If you simulate 7,621 Brownian motions,
yeah,
and do the same diffusion coefficient, what is that distribution?
Well, it's more like over here. It would depend on how long they all were and a few things, yeah, but it's definitely over here. Well, I'd have to tell you what the diffusion coefficient was.
Presumably, you could even estimate it from these tracks, like take the average of all of them.
Yeah, these are actually, because these are Q dots, these are quite long. So the average of this histogram is going to be very different than what it would be. And the average appears to be they're not moving. Or they're moving very, very little.
How do you determine the immobility threshold?
That's from that calculation which I showed you. So basically that's the point at which we can't distinguish a mobile one from a stuck one. Yeah. We have another immobility threshold, which is actually we just take the diameter of the track and we say that that can't be more than a threshold that we set, and we say we do. Does that make sense?
Is this the S squared over 2 tau thing?
Yeah, S squared over 2 tau is saying that you're just looking at noise. There's another one which is we're just going to take the whole track and we're going to say how big is that? And for stuck tracks, we're going to pick a value where 95% of the stuck tracks are identified as stuck. But that we did it the diffusion coefficient way here.
And your track, do you have a lot of redundancy in where there are recurrency? I mean,
No, not too much. Not too much. There's a lot of cells here. There's a lot of cells. Different experiment than yours.
Okay.
Right. Different experiment. There's a lot of cells here. Yeah.
Okay.
So, sorry, just to go to that last comment. So this is across the surface of many, many cells.
Many cells.
Okay.
Many cells. Yeah.
Like how many cells compared to track?
Well, there's 7,000 tracks. We get about 50 tracks per cell. So, okay. All right. So then we perturbed everything with drugs. And I've overlaid the histograms here just to make the point that there's no difference in any of the histograms as far as we can tell. So basically, we completely don't get the same results as far as the drug perturbations go. And then looking back at these, remember these thresholding pictures? We decided that we would threshold, oh shit, that's really annoying. We decided, okay, this is a movie of the threshold. Okay, but I'll just show it as a still. So we thresholded, and we tried various thresholds, and this is, and with our favorite threshold, we see no difference. We probably can, because we've got so many tracks, there probably is a statistically significant difference here, but I don't want to push that. So yeah, basically we completely failed. So we could not reproduce the actin control results. But then we looked at our tracks, and essentially very few, more than half of them are just basically not moving. And so it's indicating that there's a problem here, so we're not picking up things moving. So we decided we were going to do a comparison of Q dot versus fab labeling. Okay, so we decided we're going to go back in with a relatively small probe compared to these big fat chunky Q dots. And then we realized that we wasted 3 months because, that's okay, because if I label the exact same thing with psi3, this is the distribution of diffusion coefficients I get. Versus if I do it with Q dots, this is the distribution of diffusion coefficients.
Where's Fab, is that like Psi 3?
Fab Psi 3, yeah.
Oh, yes, I think.
Yeah, it's a small thing. It's like this. I mean, this is actually kind of a scale picture of what we think's going on. This is with IgG, which is one kind of B-cell receptor. This is with the IgM, which is a different kind, just to verify that we're seeing similar results. Okay. We
So why
check.
did we waste your time?
Well, we wasted our time because most of the Q dots it appears were just stuck. They just weren't moving. So the fraction of things which, I mean, 0.02 micrometers squared per second is quite slow, okay? Whereas in reality, we're seeing these things, and we're not doing anything to make them go faster, we just label them as something different. So really, they should be moving more like this fast, or maybe faster, but we were observing them not moving. We also looked at, remember I said the substrates were different? So we tried different substrates, and just quickly, if you just look at these 4 plots, you can see there's no difference in the substrates with Q dots or with fabs. Okay. And then we also looked at IgD, which is another kind of B-cell receptor which doesn't move, and labeling it with labeling IgM. Sorry, I'll look over here first. IgM with Psi 3 goes faster than Q dots with Psi 3. Sorry, I'm saying this wrong. This is IgM. This is IgD. These are both with Psi 3, the small label. IgD doesn't move as much. Okay. These are both with Q dots, where you would think that IgM was not moving to the same extent that IgD was not moving. So there's a biological interpretation that goes along with this, but basically it's saying that you cannot distinguish quite big differences that you can see with fabs if you use Q dots. Yeah. All right, so to summarize, I do not recommend or condone Q dot labeling. So that's actually a result. I'll take that as a result. However, if you do use Q dots, then the trends tend to be the same way. So if you do a drug perturbation experiment, you'll typically see the same trend. Q dots have their merits because you can do very long tracks. So if you're imaging on the dorsal side of the cell, so away from the glass, say in confocal, Q dots might be all right. IC beats U track, that's my opinion. And on the B-cell specific side, well, I believe that the Q dot motion is influenced by the topography of the membrane relative to the glass. So basically, when you're doing a Q dot experiment, what I think is happening is there are gaps between the cell and the glass, and the Q dot explores those. And if it's stuck away from one of those gaps, it just sits there and doesn't move. So I'm not a big fan of the Q dot turf system anymore. It seemed like such a good idea, right? They never bleach, you get very long tracks. I wrote a whole grant proposal with these things in as the #1 thing we're going to do, and now we have to change all this.
So you got funded, that's what's important.
Yeah, but I always get funded. I'm in Canada, of course I get funded, right? I didn't say that. All right, okay.
But this was not like a well-known problem with Q dots, right? So you discovered some limitation.
If it was well known, it wasn't well known to me or anybody that I heard from, and we do have. The thing is what's a bit tricky at the moment is we had a paper come out in Nature Communications that was sort of prior to all these discoveries, which was using Q dots, but the Q dots seemed to work better then. So I don't know. But in our hands, Q dots didn't work all that well. I just want to quickly take you through some new stuff. I think I've got 5 minutes left. All right, so remember BCRs, they form clusters. So this is a dead cell, but we've labeled all the BCRs on it, and it's fixed. It's a fixed cell. We've labeled the clusters of BCRs. Okay. And there's this model which is gaining popularity. Basically, when the B-cell starts to signal, what happens is the BCRs are kind of held apart by the cytoskeleton. So they're kind of in small groups, but there's like cytoskeleton in between the groups. This is the model. I'm just telling you what the model is. And then when you activate the cell, basically the cytoskeleton reorganizes and allows these small clusters of BCRs to merge together into bigger groups, and this can drive signaling. This is the model which you'll find in the literature.
Is this just thought to be a binding interaction or just a steric blocking?
Steric blocking.
So how far do the receptors penetrate into the cell?
This is very if I had to like gamble, if this model is correct, I would say there's probably secondary proteins involved in making this work, as far as pickets and fences go. Yeah. All right. So redrawing that model, there's this kind of energy diagram. Okay, so you've got kind of signaling level on the left, and then you've got If the cell's asleep, then it kind of looks like this picture where the BCRs are all held separately, and if the cell is activated by, say, a high dose of antigen, then you kind of get to here, okay? So previous work at UBC kind of showed that there might be a sort of intermediate activation step where basically you could stimulate the cell with TLR ligands. So these are like LPS, which is on the surface of bacteria. So if a B cell meets a bacterium, it will get stimulated by the LPS. Okay, it kind of puts it in an intermediate step, and sort of somewhere in between this picture and this picture, there's something like this picture, where there's kind of medium-sized. Okay, so Goldilocks, right? Okay, so you've got like, yeah, everything's separated, things are kind of mediumly separated, and then things are not separated so much. All right. Okay, so this is like a picture model from the biology. All right. So the hypothesis is in the TLR activated cells, so in these intermediate cells, BCR co-localizations that we measure should occur more frequently and last longer. Okay, so there's a problem with studying co-localization with SPT, which is if you're doing single color, it's hard to resolve the particles when they're close together. So if you're trying to resolve localizations, you start to lose your ability to determine where the particles are. You need the particles to be separated in space in order to properly track them. Okay, so the solution is, let's use two colors. So we took a B cell, we labeled the IgG on the B cell with Cy3 and 488 dyes. Okay, so some of them are red and some of them are red and some of them are green. Okay, and we watched them in two-color single particle tracking and define co-localization when they're closer together than some distance r. Okay, and we'll vary what r is. And we expect more and longer co-localizations if we make r big, right? Okay.
And we should be seeing, I mean, those two yellow circles, one will come up red and one will come up green.
Yeah, that's supposed to be red and green, but it doesn't really look like that to me.
So fret also happening here?
No, no fret. Okay. Okay. So first of all, the diffusion coefficients are independent of being in red or green. That's actually a good thing to check, and also independent of the person doing the measurement, which is another good check. And then we have the control cells, and we have the LPS treated cells, and there's a small increase in diffusion coefficient. Just purely looking at this without worrying about co-localization, there is a small increase in the diffusion coefficient between the control cells and the LPS treated cells. So this is actually a good thing. So this is all good.
It's the type of graph you'd see on Fox News to show how much debt has increased under Obama.
Yeah,
It's like
I know.
it's
I didn't
right?
actually make this
Yeah.
graph, yeah, but it's gone from gone from 0.1. Yeah, but look at the number of tracks we have. All right. So if you had that many countries, I think you might support that kind of graph.
But is that a statistically significant difference in trying to interpret your standard deviation relative to the change?
Yeah, it might not be. I don't know. Yeah, but I'm just trying to show it goes up. This is work in progress. I mean, I'm not going to publish this graph. I'm only showing it to you because it's the one that the student gave to me. All right. Yeah. All right. So now we looked at collision frequency. Okay. So what we do is we just work out how many collisions do you have per million pairs of tracks. Okay. So these are on different cells. So collisions can only happen on the same cell. All right. But we do this per pairs of tracks because that seems to be the proper way to normalize it, right? How many collisions do you have per million pairs of tracks? And basically, it kind of looks like this. And it actually looks like we get more collisions. This is a function of the diameter, which I set. We actually get more collisions, sort of by a small margin, in the untreated cells, which is a bit contrary to what we might think. So we don't currently have a good explanation for that, so I'm not going to try to give it to you. And then I can, as well as just counting how many collisions there are, I can count how long do the collisions last, co-localization, sorry, when they happen. Okay? So as a function of the expansion diameter, okay, as a function of the distance where I define a co-localization occurred, I basically get a small uptick in the, as a function of the diameter, and basically no difference between the BAF and the BAF LPS, so between the controls and the activated cells. All right, so then I asked myself very quickly, do these durations reflect non-random association? Okay, so what I can do is I can take two particles diffusing with the given diffusion coefficient, and I can work out how long should they stay together for, right, if they're just diffusing. And I know how to do that. It's a mean first passage time problem, so everybody likes this. So I solve it, and I get the mean first passage time for two particles with the diffusion coefficients that I measure should be about just over a millisecond at the biggest expansion diameter that I've been showing you. Okay, but what I've been observing is about 100 milliseconds. So there's a big separation between the theoretical time and the observed time. So basically these particles are staying together much, much longer than you would expect according to a pure diffusion model.
When you have the matrix still there, then maybe that makes sense, because then if two particles get the same pocket, they get stuck there. But again, so maybe it's more surprising when there's kind of more freed from the matrix, it's supposed to be moving more freely.
Yeah, well, I mean, we have one set of data on this. Okay, I mean, there's lots of tracks, but it's like the control and the experiment. We have one set of data, and what we're going to do now is go back and get more.
Yeah, didn't Tatiana can speak for herself, but in her talk, didn't she talk about having hierarchies of compartmentalization on the plasma membrane?
Yeah.
So it's a different scale. So you have a fine scale and a larger scale.
Yep.
Compartmentalization, and might that explain?
I like that model.
I think it's a nice idea.
I like that model. Yeah. I have no issue. Yeah, having a hierarchy of compartments would be one way of getting around this.
Yeah.
But then you'd also have to somewhat explain differential modulation of the different compartments by different stimuli. But I think that would be a great model, and if we found evidence for that, it would be a really nice thing to do. Yeah, I have to see which way the evidence points. But yeah. Yeah.
Have you looked what happened for R larger than 250 nanometers? This is very small.
No, we didn't go bigger than that, but we probably should.
The potential one we found, the smallest one are around twice the size and go up to, let's say, 800. So this is very small.
So to go
this
to
is
my-
very small.
Yeah.
I would be interested to see what happened if you can, look at...
Oh, we could. Yeah, we could.
okay.
Yeah, I think our cells are a bit smaller than yours on the whole,
Okay.
but yeah, we can go big. We can definitely go more than 250. Yeah. So this was somebody's honest thesis. Okay, so I said, okay, why don't you do 50, 100, 150, 200, 250, and then we'll stop. But we didn't think about it too much. So yeah, yeah, this is a great suggestion. Yeah, I can also calculate what I think the mean variance should be for the time to escape, and the variance is pretty close to the mean first passage time, which is what you expect. It's approximately an exponential process, blah, blah, blah. But it sort of explains why, it's just sort of further evidence that what I'm seeing is completely different than a pure diffusion model would predict. Right. So tentatively, there seems to be no difference when you stimulate with LPS. I mean, maybe there's a little bit of variation here that I can pick up. All cells show substantially longer co-localization than predicted by pure diffusion. What are we up to? We're doing more experiments. We're trying to do higher time resolution experiments. We're trying to simultaneously image clusters and the receptors. Modeling-wise, we're just slightly improving things, that there's a possibility that the thing diffuses out and comes back. We're just going to push that. That's never going to explain the difference. We're just going to push that away though. And thinking about, is what we're measuring as diffusion really cluster diffusion as opposed to particle diffusion? Okay, so with the cluster imaging, we're going to try to measure the cluster diffusion. But that's all in progress.
What's cluster diffusion?
Well, if you've got clusters, the clusters are moving around.
What's cluster imaging?
Cluster imaging means we image instead of imaging B cell receptors individually at low density, we image at high density in green, watch them move, and try to watch the greenery diffusing around in sort of a bulk way. And then we label the red ones still individually. And so we have the greens moving around in the bulk in sort of a big agglomerated way. And then we have single red ones moving around with them. So,
So models of rafts and non-raft diffusion, is that
yeah,
the sorts of thing you're
that would be
talking
the
about?
idea.
Yeah.
But it's challenging.
So,
It's challenging
it's
to
controversial as well.
Yeah, it's challenging to image the dynamic clusters, so that's something that Levin's working on right now.
And there are evidences that how the clusters are formed. Can you have also an idea of how many receptors you have in a cluster?
Yeah, we get that from Storm. So the nanoclusters, that's part of the reason we picked 250 nanometers actually, is that the nanoclusters that we see in Storm for resting cells, I mean, I don't want to go overboard, but we're guessing, a few hundred, maybe 100 nanometers in diameter, kind of spherical if they're round, and maybe 50 BCRs, maybe a bit more, you know. But that's in Storm where hopefully we're picking up everything. We're maybe not picking up quite everything, but it's not going to be a factor of 10 more. And now some philosophy, Thursday afternoon. But you know, you only have the right to do that. You have to enjoy the process. So I have to learn to enjoy the process of the experiment, like being hard to interpret, rather than worrying about what the result will be. All right, I went over, and I'm sorry about that.
So I have to leave in 5 seconds. I just want to ask this question.
You should update that.
If you have so many trajectories, even if they come from different cells, assuming that the cells are more or less homogeneous, and that the slowing down is due to aggregation, not an interaction with something underneath, like a potential well. You have, let's say, the reason for stopping or reducing diffusion is because you have this aggregation. Then that would be still interesting to assume that when we do average over all of the cells, it's like one cell, use the homogeneous approach that we've developed, try to use your data, extract diffusion coefficient locally, and then indeed check whether you still have some potential wells, because this is what we can do. In principle, we should not see this, and actually we have proved in the past using VSVG, which was a protein of the HIV virus, that you don't have potential well. This was the data of Lippincott Schwab that she published maybe 10 years ago. No potential well, no arrows converging, nothing. So then we should have the same in your data, nothing. So then what we see, what the results, the reason for having small diffusion is aggregation. And this is very nice. This is something that is, you know, a different reason of slowing down. It's by molecules coming together. And also what would be very interesting is to understand how these clusters are organized. So I think there is a possibility that we could use the methods on your data, although they are taken, I mean, you have 15, I don't know, 20 frames per cell, still it might be interesting, assuming you have homogeneity, so we can pull everything like as if it was one cell.
Yeah, I think there's something to be done. It's a distinguishing between sort of spatial and temporal regulation, I think, is what you're trying to get at, right? So you're talking about spatial regulation, which is what I was talking about at the end, right? Yeah.
Do you have a way to increase the number of trajectory per cell?
It was challenging because eventually they overlap. Like, okay, we're in red and green. We can't, if we over-label, then eventually we can't distinguish the single particles anymore. So we could increase it a bit, maybe a factor of 2, but I don't think it's not going to be like your experiment. The only way it gets like your experiment is, you remember there was 3 lasers on the microscope? So one of them is for activating the receptors for palm, and at that point, yes, right? But, good, you should go.
Okay.
Yeah, nice. So if you had a model where you have clusters, and there's a Markov process between being on a cluster and off a cluster, and you have a certain unknown diffusion rate on a cluster and a certain unknown diffusion rate off a cluster, do you think you could develop a simple enough model to?
Yeah.
Is that where you might go with that? Or?
That might be a good direction to go in. Yeah. I want to make sure if we can image the clusters first before I go crazy on that.
Yeah.
Sam?
So, like your picture of the B cell, you have all these projections coming out on the surface, right?
Oh, the EM, yeah.
So, and that's what you're diffusing around. And so I just wonder, could that have some kind of effect with causing you to see things look like they're trapped? Or?
Yeah, I think it could. Although I'll show you another EM. Hopefully it shows up. Sorry, it's not. Okay. With adherent cells, if you EM them after they've adhered, they're really pretty flat. Those B cells are just sitting there, right? So if we EM the B cell after it had after it had sort of spread itself onto the slide. I think we do have a few images like that.
I don't know what you mean by EM as a verb.
Electron microscopy. Yeah. So if we did that, then I think on the underside they are more flat, but I wouldn't swear to.
Yeah, so I was getting another question is when you're doing the imaging here, they're not in kind of their in vivo shape. They kind of
spread.
relax.
Yeah. They're more like a fried egg kind of shape. Yeah. But that's also that's an interesting thing because some of the cells like they don't stop moving, and so you never image those because they keep moving, right? So you only image the ones that have kind of stopped and hung around. Yeah. Yes.
My collaborator at Maryland, Arpidu Bhattaya, I don't know.
Does exactly stuff like this. Yeah.
Yeah, and she was looking at this at the centripetal motion of basically the active retrograde flow and the cluster motion, and I thought that I don't remember. I mean, actually, we have a proposal together, so I should know. But I thought that at least there was some correlation that she found in terms of the centripetal motions and so it's
Yeah.
maybe more.
Yeah, she has this nice biophysical paper, right? Yeah. So yes. Yeah, I was looking at it just the other day.
So would that be more like an sort of support an argument that actin is involved?
Yeah. Things wouldn't be exactly the same. So our cells are completely resting, right? I mean, she's stimulating her
Yeah,
cells.
I think she was looking at contact spreading.
Yeah.
And another issue related to that, if actin does convective motions versus diffusive, and I don't know if in the analysis that you did that you sort of picked it up that you have a convective component.
Yeah, so we did that as I didn't present that, but yeah, we did look at drift diffusion or convection diffusion as you know, so what's the maximum likelihood estimator for those things, and we've done it on simulated data and it worked well. And when we ran it on our data, it was very hard to convince ourselves that that model fit better than just a diffusion model. So that was the issue for us there. Yeah, but we haven't tried it recently, so it may work better. Yeah. Drift diffusion is a tricky one, right? Because
Yeah, but also I think that the trickiness there is that the cytoskeletal drift, it also has a characteristic correlation length and time scale. So it's not going to be a static drift diffusion where you have sort of a static convective component, but that itself on some time scale is going to vary.
That's the problem. It's going to go in different directions and stuff as well, right? So it can go a little bit straight and then a little bit straight.
So you'll need some hidden model for that, I guess.
Yeah, I'm not sure. I mean, when it comes down to it, you're just making your sample step length distribution different in some way, right? And anyway, maybe we should let people go. I feel a bit guilty.
You're just thirsty, that's all.
Huh?
You're just thirsty, that's all.
No. Okay.
Well, we
Peter.
can continue at the top.
Peter,
Yeah, no, I
sorry.
just want to
Yeah.
comment with regard to your Bhagavad Gita quote. There was a bachelor lecture in the physics department earlier this week, and the speaker quoted bachelor saying, "Work is more fun than fun."
Can we leave it at that?
Thanks, Dan.
Good.